Welcome back to Sleep Academy Online. In this class, we'll be talking about sleep biology. So what do we actually know about sleep? What do we know about sleep is that sleep is known as no longer as a dormant state. Sleep is a fundamental and highly complex biological phenomenon. Sleep has a very complex biology. So in this class, I will explain to you simply what is sleep. There are two mechanisms which control our sleep. Number one is circadian rhythm, and number two is adenosine tank. What is circadian rhythm? It's our internal body clock, which controls all our daily and nightly cycles. Basically controls the timing of our bodily functions during 24-hour cycle. As you can see on this diagram, this is a 24-hour clock diagram showing what is our body actually doing at different times of the day. And this is controlled by circadian rhythm. And adenosine is a neuromodulator produced by our cells, and it travels through our blood to our brain to attach to adenosine receptors. Before we continue about adenosine, I wanted to outline the differences between circadian rhythm versus biological clock. Our biological clock is not our circadian rhythm. Biological clock is a timing device of all cells and tissues of the body. And circadian rhythm is a natural internal process that regulates our sleep and wake cycle every 24 hours. Biological clocks produce circadian rhythms and regulates their timing. Master clock is, again, something different to biological clock and circadian rhythm. Master clock coordinates and synchronizes all biological clocks in living organisms. It's located in the brain and is called suprachiasmatic nucleus. It's located in hypothalamus and made up of 20,000 neurons. It's a very small part of hypothalamus, and compared to the rest of the body, it's very, very tiny, which only has 20,000 neurons compared to the body that has billions. So our master clock, aka suprachiasmatic nucleus, controls our circadian rhythm. However, it is influenced through our eyes by external factors, such as light and darkness. As you can see on this diagram, light comes in through our optic nerve straight into our hypothalamus, and therefore it gives signal to suprachiasmatic nucleus, and therefore suprachiasmatic nucleus sends signals to the body to control or activate specific function. It activates a lot of physiology and behavior in the body. The suprachiasmatic nucleus processes signals coming from the eyes and releases hormones and neural signals which trigger behavioral and physiological events. This could be anything from lowering the metabolism during different times of the day to making our organism more active during other times of the day. So in short, suprachiasmatic nucleus is our principal circadian pacemaker, our circadian timing system. Now back to adenosine. Adenosine is created naturally in the body. Every cell in the body contains adenosine within its DNA and RNA. Adenosine is a neuromodulator affecting the complex behavior of sleep, largely the initiation of sleep. Accumulation of adenosine in the brain inhibits wakefulness. The longer we stay awake, the more adenosine we produce. And during sleep, levels of adenosine decrease, which promotes wakefulness. So in short, when we wake up in the morning, our adenosine tank is empty. And during the day, every hour, the tank fills up with more adenosine. So we have adenosine receptors in the brain, and the brain is waiting for adenosine to attach to the receptors. Adenosine travels through the blood, and as the day progresses, we accumulate more adenosine into the tank attached to the receptors, and when the tank is full, we can then go to sleep. So if the tank isn't full, we aren't ready to go to sleep. How does caffeine fit into this? Caffeine's molecular structure is very similar to adenosine structure. So when caffeine is consumed, it blocks the action or processing of adenosine within the brain and inhibits sleep. So when we consume caffeine from coffee or black tea or green tea, or any other beverages that contain caffeine, our body is confused. So what happens is caffeine then replaces adenosine in terms of caffeine molecules attach themselves to adenosine receptors, and therefore our body can no longer accumulate adenosine in the tank because adenosine receptors are blocked with caffeine. So when we drink coffee, coffee's half-life is 6 hours. That's only half-life. So for the next 6 hours after you drank a cup of coffee, your body will be taking caffeine molecules and attaching them to adenosine receptors instead of taking adenosine to the adenosine receptors. So therefore your adenosine tank for the next 6 hours will be empty. This is why we have to be very careful with caffeine. So it's worth mentioning here that if our circadian rhythm is right and adenosine tank is full, we are ready to go to sleep. So if one of those processes is off, we are not ready to go to sleep. So now let's talk about stages of sleep. Our nightly sleep is composed of a number of sleep cycles, each of which is made up of different stages. There are two types of sleep, non-REM and REM. Each cycle of sleep in a healthy adult takes about 90 to 120 minutes. In children, it's much shorter, about 30-40 minutes, depending on the age of the child. The differences between REM and non-REM sleep, types of sleep, are almost as profound as the difference between sleep and wakefulness. So therefore, they're very different, and we'll go into depth very soon. Every night we pass through 4 stages of sleep, previously known as 5 stages in each cycle. And usually we sleep for about 4 to 5 cycles every night, or we should be sleeping through 4 to 5 cycles every night, depending on duration of your sleep and depending on duration of each cycle. Like I've mentioned previously, each cycle takes about 90 minutes, depending on what you've done during the day. It can vary from night to night. For example, times when somebody is very stressed, their sleep cycle can be much shorter, it could be 60 minutes. So let's go into those stages of sleep. Non-REM sleep is made up of, which is non-rapid eye movement sleep, is made up of 3 stages. So stage N1, light sleep. It is light sleep, typically occurring during sleep onset. So basically, N1 usually seen at the very start of the cycle, and we see it as we are falling asleep. So sleep onset is like initiation of sleep. Usually it's a theta waves and made up of 5% of total sleep duration. Stage N2 is also light sleep, but it's deeper. It's also theta waves. It takes about 50% of total sleep duration. Stage N3, previously known as stage 3 and 4, is our deep sleep, slow wave. We see here delta waves, and it takes about 20% of total sleep duration. REM stage, which is rapid eye movement, usually we see alpha and beta waves, and it takes about 25% of total sleep duration. So now let's go in depth into each stage and see its characteristics. So stage N1 is usually characterized by transition into sleep, and it's always at the start of each cycle. So it lasts about 5 minutes, and what we see is slow eyelid movements. So when we see on EEG, when the person is hooked up to monitors in the sleep lab, we see slow eyelid movements. Usually muscles are relaxing in this stage, and we can be easily woken up. So if we are in N1 and we hear noises, somebody calls our name, we will wake up quickly and easily. Stage 2 characteristics, it's also a light sleep, but this is classed as a true sleep, which lasts about 10 to 25 minutes in each cycle, depending on the cycle that you're in. And usually eye movements stop and heart rate lowers and body temperature decreases in this N2 sleep. Stage N3 characteristics is, this is a deep sleep. This is our deepest sleep that we get at night. We are not woken up very easily from the sleep. If you try to wake somebody up from deep sleep, it'll take you a while to wake them up. And usually people would wake up very confused and feeling groggy, and they don't understand where they are. So it would take them a few minutes to come back to reality and understand where they are. This is a slow brain waves, which are delta waves, and this is the almost restorative sleep. Usually blood is redirected from the brain to body to restore cells. So this is where most human growth hormone is produced, and this is where sleepwalking and other sleep related disorders can happen, such as sleep talking, non-REM sleep behaviors usually happen in deep sleep, and this is where our muscles are not paralyzed. We can walk, we can eat, we can talk in this sleep, which we will not remember the next day. Stage REM characteristics. This is called paradoxical sleep. Eyelids move rapidly, and this is the stage where we are dreaming. We can also dream in deep sleep, but we will not remember those dreams. But when we are dreaming in REM sleep, we will remember the dreams. This is where we experience muscle paralysis. Our major muscles are paralyzed at this stage, therefore protecting us from acting out our dreams. And this is, we experience a shallow breathing and our heart rate and blood pressure increase. This is our last phase in sleep cycle. This is a full cycle that we've just been through. Once this cycle is done, we actually go through very brief period of wakefulness, could be for a couple of seconds, and then we go straight back into N1. And this is the hypnogram of one sleep cycle. And this is where you see W is for being awake, R for REM sleep, N1, N2, and N3. And this is, and down on the bottom is the time. So you'll see basically how we transition from one stage to the next, and this is just one cycle. So, and as you can see, at the end of this cycle, we come back to wakefulness and then straight into N1. A lot of times when people do wake up during the night, they usually wake up after they had REM, and they wake up briefly. Some people wake up enough to go to the toilet and remember that they've been awake, but some people don't remember the transition straight into N1, and they don't remember being awake. And this is where you can see sleep stages as well. So this is the differences of sleep cycles. So each cycle that we experience during the night is different to one before it. So usually within the first cycle, we spend most of our sleeping time in deep sleep, and not much time in REM sleep. So as we continue from cycle to cycle and towards morning, we spend less time in deep sleep and more time in REM sleep. And we can also go for a few more wake kind of stages. And we might remember them, might wake up briefly and then go back to REM, and wake up and go back to REM. So we don't get as much deep sleep. However, it's normal not to get more deep sleep towards the morning. This is how we actually sleep. And this is why it's better for us to go to sleep rather earlier than too late, because we don't want to miss out on that deepest, most restorative sleep. This is where most of the human growth hormone released, because during the day we don't release human growth hormone, but only during our deepest sleep we release that human growth hormone. Therefore, this is why children, those who don't get enough deep sleep, they don't grow as well as they could if they were to sleep well and to spend more time in deep sleep. And human growth hormone also is needed for adults as well, to restore the cells and to restore our cellular processes in the body and give us that repair to the cells and tissue. This is why we do need human growth hormone. This is why this is called most restorative sleep, because human growth hormone would go and repair our tissue, our cells. So how is sleep actually measured? As you've seen before in the previous slides, how do we actually measure these stages of sleep, and how do we actually see this hypnogram? So this is what we do. We have in the sleep lab, we have EEG, which is electroencephalography, which measures brain waves. So in the lab, we hook the patients up with the wires, and we measure EEG, EOG, EMG, and other different measurements, but in here we just talk about the main ones. So basically EEG, we see all those brain waves that I've mentioned before. EOG is electrooculography. This is where we measure the eyes, so basically to understand either the person is in REM, and we do need to see REM when we're doing sleep studies in the lab. So because a lot of things could happen in REM, for example, different in breathing. So when we're watching the patient in the diagnostic study for sleep apnea, we need to see what their behavior is during REM. EMG is electromyography. It measures muscle movements. So basically 3 electrodes which are connected to the chin. So usually we measure activity or inactivity in EMG to understand what kind of stage of sleep that the person is in. For example, if it is REM, EMG movement will be non-existent, and during other stages of sleep, EMG will be active and the person might be moving the chin. So that would help us understand either they are in REM sleep or non-REM sleep. And the whole system is called polysomnography, which short as PSG. And this is full polysomnography is done in the lab and is monitored by a sleep technologist in a sleep lab all night, or there are home studies done, which is also called home polysomnography, which is not as comprehensive as one that's done in the lab. This is what the brainwaves actually look like when we look at the monitor in the lab. So as you can see, wake with mental activity, so you can see beta waves, and these are the really fast and very short kind of waves. When we see a person wake but resting, which is alpha, once the person closes their eyes, the brain goes into alpha rhythm. And when we are relaxed, we also are in alpha rhythm. And this is what alpha rhythm looks like. And when we are sleeping in a light sleep, our light sleep looks like that, which is in theta waves. And our deep sleep is a delta wave, so it looks something like that. Well, this is about 2 seconds epoch, and as you can see down bottom, the measurement of 1 second. So I think this will be a 2 second epoch. So this is what it actually would look like, something like this. And this is what electroencephalogram looks like, EEG, showing typical brain waves of sleep and wakefulness. And this is what we would see in a sleep lab, and the sleep scientists would go and actually score this, will determine how many hours or minutes a person spent in a particular phase, N1, 2, 3, or REM. So each of these lines, each of these EEG will be identified. These different stages will be identified by its own characteristics, such as spindles, K complexes, slow waves, or theta waves, or those alpha waves. So as you can see, the differences are there, and this might be just a 30-second epoch. So in the sleep lab, what we do, we measure every 30 seconds of sleep of a person during the sleep study. So we need to sit there and watch them sleep practically on the screen, and we watch which stage, and then we report. We write a report after half an hour of each sleep. We write what kind of sleep that is and which stage the person is in. And when the sleep scientist scores the study, they would have to identify how long the person spends in each stage of sleep. But the interesting part is that we don't solidly sleep for 20 minutes in REM fragments. They can go for all of the stages in those 10 minutes. So we don't sleep in big chunks. We can shift from stage to stage within a matter of minutes, and then go back to another stage, and go back to light sleep, go back to deep sleep, we can go to REM sleep. So it's not as simple as going from 20 solid minutes in one sleep and then 20 solid minutes in another sleep stage. So it's a bit more complicated, but this is what basically looks like. This would be about 30 seconds epoch. In here we can also see the stages of sleep when we are awake, because you can see beta waves. This is what it looks like, and there is a measurement of 1 second. So you can see this be about 30 seconds or even less. And drowsy, when we're drowsy, relaxed, we are in alpha waves, and that's what it looks like on our EEG. And when the stage 1, it looks a little bit slower. It's slower, but it's not as slow as deep sleep delta waves. And stage 2, we see K complexes and spindles. As you can see, they are outlined there. So this is how you identify which sleep stage the person is. And then stage 3 and 4 sleep, we spend in delta waves, which is our deeper sleep or slow sleep. And this is how it's characterized by when it looks like that. And then our REM sleep is completely different again. It's characterized by sawtooth waves and also inactivity in EMG. So that's it for today. I'll see you in the next lecture. Thank you.